So, hi everyone. The topic for today is basics of developmental biology. In this, we'll be studying about the basics of developmental biology, wherein what is developmental biology, what are the different steps involved in a developmental process of an organism throughout the lifetime. So, specifically in developmental biology, we study the series of progressive changes that generate a structure of organism throughout the lifespan. And in the progressive changes that we study about is the cellular changes on a genetic level, which involve the cell and cell interaction. So the steps basically which involved is from gametes undergo fusion to form a zygote, which eventually undergoes cell division to form a cell structure known as morula, which forms blastula. And then there's this blastula, which contains a blastomere and blastostele, which then undergoes gastrulation and forming a gastrula. So gastrulation part is the differentiation part of the blastula. This step determines which cell will produce what kind of organ system in the body. After the gastrula stage, morphogenesis occurs, which is basically the organogenesis or the organ formation of the organism. And finally, we have the organism which is fully formed. So starting from the gametes till the formation of an organism, this process, the study of this process, the progressive changes that occur in this process are known as developmental biology. So what does the development specifically include? Development includes three major things. First is the growth, in which the increase in number of cells occurs, and while the increase in number of cells happen, the size also increases. Second one is the differentiation, wherein the cellular specialization is happening. And the third one is the morphogenesis, where the body shape and the size is established. There's a phrase which goes like determination precedes differentiation. So, what is this determination? Determination is the thing where it decides that which cell will develop into what kind of organs. So this will precede differentiation. Differentiation is the result of determination, where what kind of organ is eventually formed. So determination is affected by two things. Number one is the cytoplasmic segregation, and number two is the induction. So induction is the process in which a substance or tissue influences the fate of a group of adjacent cells. So what kind of fate does the adjacent cell undergo after the tissue is there? So that is called as induction. They decide the current, just say for example, this A is one kind of cell and this is surrounded by different kind of cell. So so A will determine how these surrounding cells are, how these surrounding cells will move, or how what kind of differentiation will happen in adjacent cells. So A will be inducting the other cell surrounding it. So determination precedes differentiation, in which cell will develop into what kind of organ will be there. So commitment of a cell to a particular type of fate is determined, and it occurs by the activation of certain genes. A differentiated cell can have an influence on the nearby cells to be modified into a particular tissue, just what induction is. So for example, in frogs, the developing forebrain, it influences the formation of lenses and cornea. So the cells which form a forebrain, they rearrange themselves so that it has the influence on adjacent surface tissues, and they form the lenses and cornea. So the steps that are involved here are: number one is the determination, number two is the differentiation, and number three is the induction. So the first part of development is the fertilization. Fertilization is the generation of zygote after combining combination of the two gametes. So zygote is the first cell which is diploid, that is 2n. first step in fertilization is the contact between the sperm and the egg cell, wherein the egg pronucleus combines with the sperm pronucleus. The step two is the contact of between sperm and egg's plasma membrane. So in these two steps, the fertilization occurs. And when the contact between sperm and egg's plasma membrane happens, nucleus of sperm is slowly inserted into the egg's cytosol, and sperm does not donate any cytosol, so the entire cytosol is donated by the egg. All the cytosol present in the zygote is obtained from the egg. So the steps involved here in fertilization is the detailed steps involve first binding of sperm to the zona pellucida of the egg, which is also known as the egg membrane. Then there is an acrosomal reaction when the sperm binds to the zona pellucida, and this acrosomal reaction is triggered by the sperm's contact with eggshell, and in this case acrosomes secrete the enzymes which degrade the vitelline membrane of the egg. Then in second step, there's the sperm penetrates through the zona pellucida because sperm is in direct contact with the egg, and then it can fuse with the membrane, with that egg's membrane. Finally, the fusion of membranes of both the things happen, and the sperm pronucleus enters the egg cytoplasm. So through this, only one sperm can fertilize one egg, and this is the structure of the sperm. We have a pointy end which contains the acrosome, which is known as the acrosome, and this acrosome is filled with vesicles which contain digestive enzymes, and these digestive enzymes they digest the egg membrane. Then there's this body which contains the pronucleus of the sperm, which has to be donated to the egg. There's a neck here. Then comes the collar region, which contains the mitochondria, which provides the energy for the movement of sperm towards the egg. Finally, the flagella, which provides the chemotactic movement. In sea urchin, so chemotactic movement is defined by some receptors or secretions which are recognized, the chemical release is recognized by the sperm and they move towards that direction. For example, in sea urchin, for fertilization, binding receptors are present on egg surface and binding molecules are present on sperm. So because of these interaction, chemotactic interaction between these two, the sperm will move toward the binding receptor. Acrosomal reaction, what is acrosomal process? Acrosomal process is basically when the sperm is in vicinity of the egg membrane. Uh, in that case, the actin molecules in the head of the sperm, they rearrange themselves to form a sharp pointy end. Mammals have ZP3 receptors in the egg. Matlab basically on zona pellucida in mammals, there are three receptors, ZP1, ZP2 and ZP3. Or you can also call them as zona pellucida 1, zona pellucida 2 and zona pellucida 3. Then there's this phenomena known as polyspermy, in which sometimes the fertilization of egg can occur with more than one sperm. This might lead to a malfunctioning individual, and this needs to be avoided so that there is a proper development of organism. And polyspermy is also lethal for the embryo because there's no differentiation that will happen normally in case of polyspermy. So there are different actions that the cell carries out, that the system of our organisms carries out to block the polyspermy. So there are two ways of blocking polyspermy. The first one is the fast or transient block, which acts immediately, and the second one is the slow or permanent block, which acts after some time. So talking about the fast and transient block to polyspermy, it prevents the fusion of any other sperm after the first sperm has fused. So there won't be any other sperm which will be allowed to fuse after the fusion of the first sperm. This will happen within 1 to 3 seconds of the fertilization. It lasts up to 1 minute, and this since this is lasting up to 1 minute, so after 1 minute the permanent block has to act. How does fast block happen? To create fast block to polyspermy, a huge influx of sodium ion occurs inside the cells, due to which after the fertilization, after which the sperm has donated its pronucleus. Matlab, as soon as the sperm donates its pronucleus inside the cell, there's a huge influx of sodium ions inside the cells. This is the cause of slow block, fast block. And talking about permanent block or slow block, in this case, there is removal of any other bound sperm in case there are different sperms that are bound instead of the one. It happens after 20 to 60 seconds. It is also known as a permanent block because after this block has been applied, there is no other sperm that can interact with the egg or that can fertilize after this block has been established. So the way a slow block occurs or the permanent block occurs within the cell, it happens after the cell signals for the slow block. So the different way in which happens is the phospholipase C is activated after the influx of sodium ions. So as soon as the fast block occurs, where sodium ions, high sodium ions का influx happens, immediately that activates the phospholipase C. And phospholipase C is a membrane bound signaling molecule. This is broken into diacyl glycerol DAG and inositol triphosphate INSP3 is released. So जो diacyl glycerol है, it remains attached to the membrane. This is the two ways. It remains attached to the membrane, then they activate protein kinase C. The hydrogen ions flow outside the cell, which increase the protein synthesis, and they indicate that proper fertilization has occurred, and then the cell division takes place. In case of inositol triphosphate 3, it is released and it interacts with the endoplasmic reticulum in the egg cell, which finally binds with the calcium receptors on endoplasmic reticulum. Um, the channels open, due to which from the ER, calcium ions are released into the cytosol, and this increases the concentration of calcium ions inside the cytosol, due to which the cortical granules are released, which block the polyspermy, and they form a hard covering out on the cell eggshell membrane. So the hard egg membrane is due to the cortical granules released. So cortical granules contain specific enzymes that block polyspermy, and uh, the binding of calcium receptors on endoplasmic reticulum occurs through calcium channels, due to which they can travel in and out of the endoplasmic reticulum, and this influx of calcium and sodium ions changes the membrane potential of the egg. So this is how both fast and slow block happen. High concentration of sodium and calcium ions within egg will lead to changes in the membrane potential, which eventually will change the configuration of the egg, and this will lead to the avoidance of polyspermy. The resting membrane potential is minus 70 millivolts, and upon if okay, so generally the resting potential of egg is equivalent to the resting potential of our muscles, which is minus 70 millivolts. When the sperms are added, there is rapid increase in the sodium and calcium ion concentration, due to which the potential increases to 0 millivolts. And even at 0 millivolts, even then the sperm can still fuse. So we need to increase the membrane potential even more, so that the sperm cannot fuse with the egg. So high influx of sodium and calcium ions is carried out, due to which membrane potential rises to about plus 20 to plus 50 millivolts. At this potential, the sperm cannot fuse with the egg. So the very rapid process of increase in membrane potential is the main criteria for blocking polyspermy in the egg cell. The next step is the cleavage and furrowing. Cleavage is basically the rapid cell division. In this case, the cells are kept together by some surface receptors, which are called CAMs and cadherins molecules. Um, the division in this case, cleavage case, can be vertical or horizontal depending on the requirement. So initial cleavage is, so if we have zygote. For the zygote, the initial cleavage is vertical. When the blastomere occurs, in that also the second cleavage is also vertical. And then after the blastomeres, they undergo horizontal cleavage. So this is how the cleavage occurs. In early blastomeres, cell divides so fast during morulation or cleavage stage because they have only two stages of division. One is the mitotic phase and other one is the S phase. And they don't have any other phases of cell division cycle. That's why the cleavage occurs very fastly. And no other growth phases are present, and cells rely on the stored material for their division. if talking about early blastomere, CDC are basically cell division cycle proteins. So cyclin synthesis occurs, which goes and takes it into the mitosis cycle, and cyclin B and CDC2 are present as which help in positive during mitosis phase. Active CDC2 protein kinase is the enzyme which is involved in this process. Post that, cyclin degradation occurs, wherein it enters the S phase and CDC2 synthesis occurs, and the protein kinase is inactive in this process. In this case, the DNA synthesis occurs. So this is the cycle that keeps on following in the early blastomere phase. Cyclin B and CDC2 are the mitosis promoting factor. We also call them MPF. Uh, these are also known as mitosis promoting complex, cell division cyclone protein. Rapid M and S phase keep on happening in early blastomere stage during cleavage, and after the activation of mitosis promoting factor, the controlled cleavage starts occurring. Mid-Blastula transition is basically the cells transit from M and S phase cycle to the normal cell cycle division, which includes all the four phases of the cell cycle. For transition from blastula to gastrula stage, the cell requires normal cell cycle, and therefore this mid-blastula transition has to occur so that the cell can go into the normal cell cycle division phases. Microfilaments are, or the contractile ring is required for microtubules. Microfilament or a contractile ring which is required for the rapid division in the process of cleavage of the zygote. Microfilament are basically contractile rings. They are present at the portion of the furrow, the cleavage portion of the furrow. These filaments or these contractile rings, they hold the cell close to each other after division. And this, how do they hold the cells close to each other even after division? It happens by the interaction of cell surface receptors of the divided cells. There are different types of cleavage patterns. First one is the isolecithal, or also known as complete cleavage. This will be carried out where cytosol is separated both ways and same amount of cytosol goes into the daughter cells, both the daughter cells. Yolk is evenly distributed within the new daughter cells. There are three types of isolecithal cleavage. The first one is the radial, second one is the rotational, and third one is the discoidal. Okay. In both radial and rotational cleavage are complete cleavage, and discoidal is the incomplete cleavage. In radial, the cleavage either occurs in vertical or horizontal patterns. Simple plane of cleavage is the radial cleavage. Example of radial cleavage is Amphioxus or frog. In case of rotational, the axis of cleavage is rotated. For example, mammals. The discoidal cleavage happens in fishes, reptiles, birds, and the yolk-rich region is present. Cleavage starts occurring on top of it. So this is the way cellular region above the yolk gets cleaved. The second one is the CentralAcetyl cleavage, in which the cytosol remains in the center and all the cells are formed surrounding the region. Example, Drosophila. In this case, yolk is present in the center of the egg. In each case, the cleavage will not occur where yolk is present, and cleavage will begin from the opposite side of wherever the yolk is present. Things which control the pattern of cleavage include amount and distribution of yolk in the egg cell, basically wherever the yolk is present or distributed. And the second factor that controls the pattern is factors in egg that influence the angle and timing of the mitotic spindle formation. So angle of at which microtubules will arrange themselves will determine the pattern of cleavage. In case vertical cleavage is meridional cleavage and horizontal cleavage is equatorial cleavage. So there are Based on the type of cell division, cleavage is of two types, either synchronous or asynchronous. In case of synchronous, even cell stages are generated. From one cell, two will be generated, then further 4, 8, 16, 32, and so on. For example, in case of sea urchin, the cleavage is synchronous. In case of asynchronous, odd number cell stages are frequent. In case of rotational cleavage, the plane of cleavage changes at every step of the cleavage. So morula, which is a ball of cells, is organized into blastula, which is a hollow sphere of cells. And there's a inner pore, which is known as blastocele, and blastocele is a fluid-filled cavity. Blastomere is the cells of the blastula. Then we move on to the mid-blastula transition. Mid-blastula transition is basically where growth phase is added to the cell cycle. Initially, the cycle include the M phase and the S phase, and after the embryo starts transcribing its during the mid-blastula transition, the embryo starts transcribing its own RNA, and this cells start moving around in preparation for gastrulation, where all the types of all four cell cycle phases are there. The mid-blastula transition will depend on the NC ratio. NC ratio is the nucleus to cytoplasmic ratio. So pre-cleavage zygote undergoes 4 division and it forms a 16 cells thing. In this case, the NC ratio is near about 0. And when the NC ratio is approximately 0, the transition will not occur. After 4 cell divisions, the or after mid blastula stage, the NC ratio is equivalent to 1, approximately about 1. So when the nucleus to cytoplasmic ratio is around 1, the mid blastula transition will occur. So this is how the nucleus to cytoplasmic ratio will determine the mid-blastula transition, and not this mid-blastula transition won't occur at a particular stage, but it will occur when the nucleus to cytoplasmic ratio is uh, equivalent to 1. It permits the mid-blastula transition. Cell surface receptors, usually they are made up of proteoglycans. They help in cell-cell interaction, which keeps the cell together. CAMs are the cell adhesion molecules. They influence the blastula formation. And CAMs, cell adhesion molecules, are the transmembrane cell surface proteins that bind to CAMs on the other cells. They are involved in cell migration and producing stable tissue structure. Embryogenesis starts from animal pole and gastrulation starts from vegetal pole. Cadherins are types of cell adhesion molecules which stabilize the frog blastula. They are known as the E-cadherins basically. Compaction requires E-cadherin. Blocking the cadherin receptor with anti-cadherin antibodies, the cell can be easily distinguished even though they are not working as a unit. Then moving on to gastrulation. In this case, the germ layers are produced. 3 different layers of the cell, which is the ectoderm, mesoderm, and endoderm. These are produced in the process of gastrulation. The During gastrulation, the folding of blastula sheets occur, which forms the 3 different layers. The process of gastrulation involves cellular movement, which forms 3 tissue layers and distinct body axis. It always starts from vegetal pole. Gastrulation always starts from vegetal pole. As you can see, this is the blastocele, this is the blastula stage, and this is the animal pole, and this is the vegetal pole. In this case, blastopore starts to form by the invagination of the cells from the vegetal hemisphere, and it keeps on invaginating. The invagination keeps on happening, and the blastopore increases in size. Ultimately, the blastophore will form either the mouth or the anus depending on the organism for which the gastrulation is happening. The archenteron forms the primitive gut of the of the organism. Ectoderm forms the nervous system and skin. Endoderm forms the lining of digestive and respiratory tracts. Mesoderms form the blood vessels, muscle, bones, liver, and heart. The cytoskeletal components directing invagination are the microfilaments and microtubules. So the microtubules get rearranged and when rearranged as and when the invagination has to occur. So this is it for the basics of developmental uh, biology. So I hope it helps. Thank you.